lab. So let's go ahead and dive in to see exactly how we do this Chemstrip urinalysis. But before we do that, this chart on page 69 of your lab manual shows some of the things that you might find when doing a urinalysis. And it also shows us some things that could cause what we're seeing. So over here on the left, this is what we might find on our Chemstrip. And then the next two columns, we have non-pathological causes. So things that could be causing this that does not mean a disease state or a disorder of some sort. And then all the way on the right, we have pathological causes. So things that could be going wrong with your body, causing us to see these results. So first, we see protein that we might find in the urine. And when we find protein in the urine, the term for that is proteinuria. Some non-pathological causes from that could be pregnancy. This is actually the way pregnancy tests work. Pregnancy tests check for protein in the urine. They're just more specific. They're looking for certain proteins in the urine. When we do this in person, I always like to kind of joke around and I ask everyone in the class who found protein in their urine, and several students will put their hands up. And then I will say, okay, which female students have found protein in their urine, and they'll keep their hands up, and then I'll end up with asking, okay, who has a boyfriend and found protein in their urine, and ask how long it's been since they've taken a pregnancy test, and that always gives some people a little scare. But you might also find protein in the urine if you have a high protein diet, because if you eat a lot of protein, then it might still show up as traces in the urine. Pathological causes we might have for finding protein in the urine: damage to the glomerular membrane. Remember, typically proteins don't get filtered for the most part. They're much too large to pass through that glomerular membrane, but if that membrane is damaged, if those fenestration slits are torn, for example, if they're bigger, then we might find some protein in the urine. Next, urea. Well, we should find some urea in the urine. Urea is a product or a byproduct of protein metabolism, and it does show up in the urine naturally, but only a certain amount. If we find too much urea, a non-pathological cause for that could be just, again, high protein intake. If you're eating more protein, then you'll have more urea show up. But another, a pathological cause could be impaired renal function. Maybe we're not, maybe we don't have good functions occurring in our nephrons, and we're not reabsorbing the urea, because a lot of that urea does get reabsorbed by our body, which is discussed in lecture. Next, glucose, glycosuria or glucosuria. Some reasons that we could find that: high sugar intake. Well, remember, 100% of the glucose that is filtered should be reabsorbed into PCT. But if you're eating a lot of sugar, then we exceed transport maximum, and it shows up in the urine. This is also a sign of diabetes mellitus. If you have diabetes mellitus, then your body is not absorbing the glucose into the cells for use. And so there's more than normal glucose in the blood, which means more than normal glucose in the filtrate, and again, we exceed transport maximum. We may find ketones in the urine, ketonuria. This could be the result of a low carbohydrate diet. If you're not eating enough carbohydrates, then your body might result to breaking down fats or even muscle, and we can see ketones building up in the blood. Some diet fads, like to do ketogenic diets, those, even though they're very popular, they are not very good for you, because again, you're breaking down fats and protein. Yeah, it's good to break down fat, but you could also be breaking down protein like muscle. And something a lot of people don't recommend or don't realize is that the brain actually can't use ketones as an energy source, so you're actually depriving the brain of some nutrients that it needs. Some pathological causes for ketonuria: diabetic acidosis or diabetic ketoacidosis. Again, if someone has diabetes mellitus, their cells don't have access to glucose, so they begin resorting to breaking down fats and proteins in the body under the impression that your cells are starving. And to tie that in, also starvation. Obviously, if you're not getting enough nutrients, if you're not eating enough, then your body will start to turn to some of its own tissues as an energy source. Blood in the urine, hematuria. This could be the result of menstrual contamination. This is usually the case in females if they do this urinalysis and they find it. Typically, when you're on your period of your menstrual cycle, you will find blood traces in the urine. But it could also, as a pathological cause, be the result of a urinary tract infection, kidney stones, which cause trauma along the urinary tract, renal disease, or such as a trauma like a blow to the kidney, something that causes bleeding in the kidney itself. Leukocytes, white blood cells in the urine, called pyuria. Again, in females, contamination from vaginal discharge is common. Pathological causes, urinary tract infection. Well, if you have an infection, you're going to have leukocytosis. So you could have white blood cells showing up in the urine, or an infection called glomerulonephritis. Nitrites in the urine. No non-pathological causes, but a pathological cause could be a urinary tract infection from certain types of bacteria. Urea, that's a tongue twister. Small amounts are normal. We expect to find some of this in the urine, but elevated pathological causes could be liver disease or hemolytic anemia. Bilirubin in the urine is bilirubinuria. Again, no non-pathological causes. Pathological causes could be, again, liver disorders or an obstructed bile duct. The last two are just telling us the pH of the urine and the specific gravity of the urine. So the pH of urine should be anywhere between 4.5 to 8.0. So it's got a very broad range. If it's a little more acidic, that could be due to high protein intake. What is protein made of? Amino acids. Acids could make your urine a little bit more acidic. Pathological causes, diabetic ketoacidosis, or starvation. If it's more on the alkaline side, if the pH is a little higher, that could be due to vegetarian diet, or pathologically it could be metabolic alkalosis, a big topic when we get to acid-base pH maintenance of the body in lecture. The specific gravity. So really what specific gravity is, it's almost like concentration. It's telling us how much stuff is dissolved in the liquid of the urine. It's telling us the concentration of the urine. And as just kind of a guideline, deionized water, so this is pure water, has a pH of 1.000. If you were to urinate pure water with nothing in it except for hydrogen and oxygen, well, we would expect something like 1.000, but that doesn't happen. There's always going to be something dissolved in it. So the normal range for specific gravity for urine is 1.001 to 1.035. The higher the number, the more concentrated it is, the more substances are dissolved in the liquid. So what we see here is I have performed a Chemstrip analysis on myself, and what we see is the actual strip that has had urine placed on each of these squares. This is what you would have done in class yourself. And then when we take that strip and we apply urine to it, we let it sit for a minute, and then there's these boxes on the bottle that the strips come in. And you line it up. You hold the strip handle up at the top, and then all of these will align. Now, just in this picture, it's hard to see. They're not completely aligned. And then I've rotated the bottle so that we can again see from this side. And now we're just going to go through and read each of the items from this chart on page 72 of your textbook. It doesn't require us to look at every single one of these, just the ones that are on this chart. So let's go ahead and, using my Chemstrip here, let's go ahead and fill out this chart. First, we're going to look at the white blood cells, leukocytes. So that is this box. And if we look, it matches with this box. And it looks negative, so I'm going to go ahead and write that in the box over here. Negative, if we look in this box here, that's normal. So you should not expect to find any leukocytes, any white blood cells in the urine, and luckily I did not. Next, we're going to look at the nitrites. So nitrites is this box right here. And it looks negative, so I'm going to write that in. And again, nitrites, negative is normal. That's what we would hope to find. Now, pH. So pH is this box right here, and it looks closest to 5, maybe 5.5, kind of in between 5 and 6. So I'm going to write 5 point something. So 5 point something, and that's within the normal range. Next is protein, specifically albumin. So that is this box, and again, it looks negative. So that's what we would hope to see. There should not be any protein in the urine. Next is glucose, this box, and it looks normal. So normal, and remember from our other chart, normal is negative. There should not be any. I actually almost expected a little bit because I think I'm pre-diabetic, so I kind of would have expected to see a little bit of glucose there, but I haven't eaten much today, so that's probably normal. All right, ketones are boxed right here. It doesn't really match the negative. It doesn't seem to really match any of those boxes. But I would say of those, it's closest to negative. So that's what I will call it. Next, urobilinogen seems to match this box. And again, that is normal, and normal is negative, so I'm going to write negative there. Bilirubin is negative, and that's what we should expect to find. Blood hemoglobin is there, and that is negative, and that's what we should expect to find. So now the box that we did not use is this top box, and the top box, we can't really see it, but right there, that box is for specific gravity. And the reason that we didn't use that box is because in lab we actually have a device that measures specific gravity. You put a drop of urine on it and hold it up to your eye and look through, and there's a chart inside, and there's a line that passes through some numbers, and depending on which numbers it passes through, that tells you the specific gravity of the liquid that you placed in the device. It's called a refractometer. But let's go ahead, and even though it's not on this chart, let's add it in. Let's add in specific gravity, since we can't do the measurement with the spectrometer, with the refractometer, and it looks like it's going to be down towards the other end, so let's use this side. It looks about right there. Now the refractometer is a lot more specific. It can be very, very specific with the numbers. This method, not so much. So it looks like it's between 1.2 and 1.25. So let's just go ahead and call it 1.22. And specific gravity, if we go back and look at our other chart, remember that is a normal, so it's within the normal range.